Oh, didn't realize you were there. All that pillaging and plundering has made me quite hungry. Let's check out Davy Jones's candy locker. In this sketch, we'll talk about gluconeogenesis. Gluconeogenesis is a metabolic pathway that makes glucose from non-carbohydrate precursors. And what better way to show gluconeogenesis than going to a pirate candy store? And not just any pirate candy store, but one with all those classic characters from Pirates of the Caribbean. The purpose of gluconeogenesis is to maintain normal blood glucose levels in a fasting state. Any metabolic disease that affects the major gluconeogenesis enzymes can cause hypoglycemia because we can't make glucose from anything that's not a carbohydrate. Lactate, derived from anaerobic metabolism, alanine, glutamine, derived from amino acid catabolism, and glycerol, derived from triglyceride catabolism, are the main substrates for gluconeogenesis. The spoiled milk, aloe juice, and igloo pie represent lactate, alanine, and glutamine respectively. Lactate and alanine are directly converted to pyruvate. Glutamine enters gluconeogenesis via alpha-ketoglutarate, a TCA cycle intermediate. Just a side note, all TCA cycle intermediates can turn into glucose except for acetyl-CoA. We drew glacier mint gum behind the DHAP treasure chest to show you that glycerol enters gluconeogenesis via DHAP. All amino acids except for leucine and lysine can turn into glucose, as you can see in this diagram. Some of the amino acids are directly converted to pyruvate, while others are converted to any of the TCA cycle intermediates, except for acetyl-CoA, of course. But all roads lead to glucose. Odd-chain fatty acids can also turn into glucose via succinyl-CoA. Even-chain fatty acids can't convert into glucose because they can only produce acetyl-CoA. Please watch our fatty acid oxidation sketch to learn more. Let's take a look at the kid wearing a pirate hat. That's pyruvate, and he's holding a bag of lactate duds. Note the towel soaked with spoiled milk. The lactate duds in towel represent lactate dehydrogenase. When oxygen is not available, like during strenuous exercise, skeletal muscle produces lactate, which gets transported to the liver. Lactate dehydrogenase turns lactate to pyruvate. The pyruvate kid is also holding a box of malt balls. The last three letters, ALT, is short for alanine transaminase. When glucose is unavailable, skeletal muscle proteins are broken down into amino acids. They're transported to the liver as alanine. Alanine transaminase converts alanine to pyruvate, which is used to make glucose. Gluconeogenesis occurs mostly in the liver, but also in the cortex of the kidneys and small intestinal epithelium to a lesser extent. Gluconeogenesis can't happen in skeletal muscle because it lacks the enzyme glucose-6-phosphatase, which we'll talk about later in this sketch. After a prolonged fast, blood glucose levels drop and glucagon is released from the alpha cells of the pancreas to stimulate gluconeogenesis. This liver-shaped candy chest containing glucagon sugar-free candy will help you remember that the liver is the main site for gluconeogenesis, and low blood glucose levels stimulate the release of glucagon. You should know that gluconeogenesis occurs in multiple parts of the cell, unlike glycolysis, which occurs specifically in the cytosol. The first part takes place in mitochondria, then the cytosol, and finally, the last step is in the smooth endoplasmic reticulum. Alright, so what are the steps of gluconeogenesis? If you guess the same steps of glycolysis, but in the opposite direction, you are close, but wrong. Each irreversible step in glycolysis requires a different path during gluconeogenesis. This diagram looks very daunting, but don't worry because we'll walk you through each important step. Pause and rewind the video if you need to. Okay, let's dive right in. First, pyruvate is transported from the cytosol to the mitochondrial matrix. You'll see another pyruvate kid walking into the mitochondria area. Perhaps he wants some Oaxacan hot chocolate? That's oxaloacetate, an intermediate of the TCA cycle. Pyruvate carboxylase adds a CO2 group to 3-carbon pyruvate, making 4-carbon oxaloacetate. We drew a forklift carrying cardboard boxes to symbolize pyruvate carboxylase. Note the black smoke emanating from the forklift and going into the pyruvate kid's face. The black smoke represents CO2. This irreversible step requires ATP and biotin, aka vitamin B7. This model B7 forklift runs on 3 P batteries. Acetyl-CoA is an allosteric activator of pyruvate carboxylase. This will be represented by the forklift driver drinking acetyl cola. When acetyl-CoA levels are already high, there's plenty of energy available inside cells, and we don't need to make more acetyl-CoA from pyruvate. As a result, the excess pyruvate can be used for energy-consuming processes, like making glucose. Oxaloacetate cannot cross the inner mitochondrial membrane and needs to be converted to malate in order to be transported to the cytosol. Malate dehydrogenase, or MDH, uses NADH to turn oxaloacetate into malate. This candy shop worker is wiping a spill from the malfunctioning drink machine. He just drank some NADH energy. The malfunctioning machine and towel will help you remember malate dehydrogenase, and the empty NAD+ bottle will help you remember that we produce NAD+. Malate exits the mitochondria and enters the cytosol via malate transporter protein. In the cytosol, malate dehydrogenase turns malate back into oxaloacetate, regenerating NADH from NAD+. Malate dehydrogenase producing oxaloacetate is represented by another malfunctioning machine, towel, and spilled Oaxacan hot chocolate. You'll also see an NADH energy drink because we produce one NADH. In the next step, PEP carboxykinase uses GTP to remove a CO2 group from oxaloacetate, producing PEP. This peppy candy store worker holding a cardboard box with kind heart candies is giving out free samples. She's wearing a kind heart button, so you know she's a kinase. PEP carboxykinase, to be exact. She's clearly gunning for the assistant manager position with her 37 pieces of flair, some lit up by 3 G-brand P batteries. Get it, girl. Note the black smoke emanating from the Oaxacan hot chocolate machine, because we lose carbon dioxide to make PEP. By the way, this step is irreversible. The next step should be very familiar if you watch the glycolysis videos. Our peppy PEP pirate will undergo the same reactions by the same glycolysis enzymes, but in the opposite direction to eventually make F16BP. Let's briefly skim through these steps. Enolase turns PEP to 2PG. Phosphoglycerate mutase turns 2PG to 3PG. Phosphoglycerate kinase uses ATP to turn 3PG into 1,3-BPG. Glyceraldehyde 3-phosphate dehydrogenase uses NADH to turn 1,3-BPG into G3P. Triose phosphate isomerase turns G3P into DHAP, and aldolase combines G3P and DHAP, making F16BP. Whew! If you don't remember these steps, please watch our glycolysis videos. They're pretty great. In the next important regulatory step, fructose bisphosphatase 1, or FBPase1, irreversibly removes a phosphate group from F16BP to make F6P. This rascal kid using a water gun to shoot the P battery out of the one peg leg of the F16BP pirate is FBPase1. You should know that FBPase1 opposes the glycolysis enzyme PFK1. Any inhibitors of FBPase1 will activate PFK1, and any activators of FBPase1 will inhibit PFK1. F26BP inhibits FBPase1, as represented by the 2,6 fruit falling on the kid's head. AMP and ADP also inhibit FBPase1. The kid accidentally knocked down an AMP jukebox and 2 P batteries, and now they're about to fall on his head. Karma's a ain't it? AMP and ADP levels increase inside cells when ATP gets depleted. High AMP and ADP means there's not sufficient energy available, and we need to make more ATP. Therefore, any metabolic processes that consume a lot of energy, like gluconeogenesis, will be inhibited, and aerobic metabolism will ramp up to generate more ATP. Citrate, an intermediate of the TCA cycle, activates FBPase1. When citrate levels are high, there's a lot of products of glycolysis present, and the TCA cycle is backed up. In other words, there's plenty of energy available inside cells, so we can slow down glycolysis and speed up gluconeogenesis. The rascal kid is shown eating orange candy, and it's got him all hyped up. Just a side note, we highly recommend you watch the regulation of PFK1 and FBPase1 mini-sketch because we couldn't cover everything in this sketch. The fruit pirate with the P battery in his 6 hook arm is F6P. Phosphoglucose isomerase catalyzes the second to last step, converting F6P to glucose 6-phosphate, or G6P. The last step of gluconeogenesis takes place in the smooth endoplasmic reticulum, as shown by the unique floor pattern. Glucose 6-phosphatase irreversibly removes a phosphate group from G6P to make glucose. We drew another rapscallion kid shooting a P battery out of the glucose pirate's 6 hook arm. Congratulations, we finally produced glucose. Yay! Final point. 4 ATP, 2 GTP, and 2 NADH are needed to turn 2 pyruvate into 1 glucose via gluconeogenesis. That's 2 ATP from pyruvate carboxylase, 2 GTP from PEP carboxykinase, 2 ATP from phosphoglycerate kinase, and 2 NADH from glyceraldehyde 3-phosphate dehydrogenase. That's approximately 11 ATP equivalents if we assume 1 NADH is equal to 2.5 ATP. That's quite expensive from an energy-consuming standpoint. But in the grand scheme of things, that glucose molecule will produce approximately 32 ATP equivalents through aerobic metabolism, so it's not too bad. Okay, let's wrap up this sketch. The purpose of gluconeogenesis is to maintain normal blood glucose levels. The main substrates for gluconeogenesis are lactate, glycerol, alanine, and glutamine. Other glucogenic amino acids and odd-chain fatty acids can also be converted to glucose. Lactate and alanine are directly converted to pyruvate via lactate dehydrogenase and alanine transaminase, respectively. The liver is the main site for gluconeogenesis. Low blood glucose levels stimulate the release of glucagon. Gluconeogenesis occurs in the mitochondria, cytoplasm, and smooth endoplasmic reticulum. First, pyruvate is transported into the mitochondrial matrix. Next, pyruvate carboxylase adds a CO2 group to make oxaloacetate from pyruvate. This step requires biotin and ATP. Acetyl-CoA activates pyruvate carboxylase. Then, oxaloacetate is converted into malate via malate dehydrogenase. Malate exits the mitochondria and gets turned back into oxaloacetate. Afterward, PEP carboxykinase uses GTP to turn oxaloacetate into PEP. PEP undergoes a series of chemical reactions in the opposite direction by the same glycolysis enzymes to eventually make F16BP. Then, FBPase1 removes the phosphate group from F16BP to make F6P. F26BP and AMP-ADP inhibit FBPase1, while citrate activates it. Then, phosphoglucose isomerase transforms F6P to G6P. Finally, glucose 6-phosphatase, which is present in the smooth endoplasmic reticulum, turns G6P into glucose.